Hello everyone. In this video, we will talk about the tryptophan operon, which is also known as the trypoperon. Along with that, we will talk about the trypoperon regulation and the attenuation mediated regulation of this trypoperon. So let's start with the video. So the first question comes is, what is an operon? So operons are the clusters of structural genes, which means that these genes together are in a group, which are under the control of a single operator site and a regulator gene, which when works together ensures that the expression of these structural genes is coordinately controlled. The TRIP operon is a type of this operon, which is a prokaryotic gene regulation process which is related with the synthesis of tryptophan amino acid. And as we all know, that the tryptophan is a very essential amino acid for any organism, be it any bacteria and all. And being an essential amino acid makes this tryptophan an integral part of so many proteins that these organisms require to function properly. So for example, if the tryptophan is present in a good amount in the environment, in adequate amount, then these bacteria doesn't need to synthesize it. So the transcription of the genes in the trypoperon is switched off. But if the amount of the tryptophan in the environment, in the surrounding is low than the required amount, on the other hand, the operon is switched on and the genes are transcribed, biosynthetic enzymes are made, and the more tryptophan amino acid is produced by the bacteria. Now, like I've already mentioned, that the trypoperon is a prokaryotic gene regulation process. So it's found in E. coli bacteria and is a group of genes that encodes the biosynthetic enzymes for the amino acid tryptophan. Now, the trypoperon regulation is completely and fully dependent on the levels of the tryptophan in the surrounding and in the environment of the organism. For example, if the amount of the tryptophan is low in the surrounding, then the trypoperon is expressed, which is being called turned on. But if the amount of the tryptophan is high in the environment, then the trypoperon is repressed, which is being called turned off. The tryptophan biosynthesis is also regulated by a process called attenuation, which is based on the coupling of both transcription and translation process. The trypoperon is also regulated by the TRYP repressor, which when binds to the tryptophan amino acids blocks the expression of the operon. As we have already talked about the basics of the trypoperon, like it's found in bacteria such as E. coli, it is required by the E. coli to survive in the environment, then it is required for the regulation of the amino acid tryptophan, then it is regulated by the attenuation method. Now, since it's an operon, it is definite that it will be having many structural genes as per the definition of the operon suggests cluster of structural genes. So these structural genes ultimately help to produce the tryptophan inside the cell, which makes the trypoperon an anabolic pathway. And the definition of the anabolism is the synthesis of complex molecules in the living organisms. So since in the trypoperon, the tryptophan is being synthesized, thus the trypoperon is an anabolic pathway. So all these five structural genes ultimately code for proteins, which in result codes for the amino acid tryptophan. So all these five structural genes are under the one promoter called the TRYP promoter. Now later in this video, we will talk about the structure of the trypoperon and we will look into the depth of these five structural genes. Now in this slide, you can see the structure of the trypoperon. So from the chromosome of the E. coli bacteria, there is a horizontal stretch taken that contains the trypoperon structure. So in this, you can see a structure called the operator where the repressor binds. The upstream of the operator contains a structure called the promoter where the RNA polymerase enzyme binds. The downstream of the operator contains a stretch of a DNA sequence called the leader that codes for the mRNA, which is also known as the leader region sequence. Downstream of the leader contains five structural elements or structural genes, we say, which codes for five different mRNAs, which in turn produces five different proteins that eventually codes for amino acid tryptophan. So these five structural genes called the TRYP-E, TRYP-D, TRYP-C, TRYP-B and TRYP-A are very essential for the tryptophan production, which we will be talking in the next slide in detail. So now talking about the genes. So there are 5 structural genes as we all know. So the first one is TRYPE that encodes the enzyme anthranilate synthase that produces anthranilate in result. The second one is TRYPD that cooperates with the TRYPE structural gene and encodes the enzyme anthranilate synthase 2 that in result produces the anthranilate as well. The third one is TRYPC that produces phosphoribosyl anthranilate isomerase enzyme. The fourth one and the fifth one are TRYPB and TRYPA that forms the two subunits of tryptophan synthetase enzyme, whereas the TRYPA makes the alpha subunit of tryptophan synthetase, the TRYPB makes the beta subunit. And these both TRYPA and TRYPB genes combine with the product of the TRYPC, that is the isomerase, with serine to produces tryptophan end result. So these all 5 structural genes only make the biosynthesis of the tryptophan possible in all ways. Now talking about turning the tryptophan on and off, as we know that turning the tryptophan on and off totally depends on the levels of the tryptophan amino acid in the environment or in the surrounding. So if the amount of the tryptophan is high in the environment, the tryptophan has to be turned off, that is called being repressed. Whereas if the amount of the tryptophan is on the lower side or in the adequate side, the tryptophan has to be turned on, that is called being expressed. So how it happens actually? So first we'll talk about it, then we'll look at the diagrammatic representation of it. So some base pairs upstream of the promoter region where we know that the RNA polymerase comes and binds, there is another gene called the repressor gene. So this gene when transcribes and translates produces the repressor molecule called the trip repressor. So this molecule is produced continuously without any stopping off, without any lagging in between, which makes this molecule an aporepressor, which means that it is produced continuously. And generally this molecule does not interact with the operators and promoters. Since the operator is not bound to the repressor at the particular moment, which makes the operator available for the RNA polymerase enzyme to come and bind to the promoter region here, and this transcribes the all the 5 structural genes that are present there. So this makes the promoter always turned on by default. It is turned on because tryptophan being an essential amino acid is required by an organism most of the times. So even though there is a repressor, it is structured in such a way that it does not affect the operon being turned on. Now in this slide we can see that the amount of the tryptophan is high in the environment. Here the RNA polymerase enzyme is present, promoter and the operator region is present along with the 5 structural genes. As we all know that some base pairs upstream of the promoter region, there is the repressor, trip repressor region is present. So now being the tryptophan amount high in the environment, the tryptophan has to be turned off. So how it actually happens? So some tryptophan binds to the specific tryptophan binding site on the repressor. It goes and binds to the repressor, trip repressor. Now once bound, it modifies the repressor structure in such a way that the repressor along with the tryptophan comes and binds to the operator structure. Now the aporepressor becomes activated because of the presence of the tryptophan, and it eventually blocks the RNA polymerase movement through the genes. Now the RNA polymerase cannot come and bind to the promoter region, and eventually it does not get able to transcribe and translate the all structural genes, which eventually turns off the tryptophan. So we can say that the concentration of the tryptophan is auto-regulating the synthesis of the trip here, which makes it a self-regulatory pathway. Now what happens if the tryptophan amount is low in the environment? So in this system, the trip repressor acts both as a sensor and a switch. It senses whether the tryptophan is already present at high levels. If so, it switches off the tryptophan. But if not, the RNA polymerase comes and binds to the promoter region, and it transcribes and translates all the structural genes, which eventually produces different proteins to code for the tryptophan amino acid. So if the tryptophan amount is adequate or is in the lower side, the production of the tryptophan happens as simple as that. So after discussing the repressor or repressing structure and talking about how to turn off and turn on the trip operon, the next topic is attenuation. So like the regulation by the trip repressor, attenuation is a mechanism, like in the starting slides we have talked about, is a mechanism by coupling the both translation and the transcription structures for reducing the expression of the trip operon where the levels of the tryptophan are high. However, rather than blocking initiation of the transcription, attenuation prevents completion of the transcription. So when the levels of tryptophan are high, attenuation causes RNA polymerase to stop prematurely when it's transcribing the trip operon. Only a short stubby mRNA is made, one that does not encode any tryptophan biosynthesis enzyme. So attenuation works from mechanism that depends on coupling the translation of an mRNA that is still in the process of being transcribed. Therefore, in this condition, attenuation actually causes or actually stops the RNA polymerase enzyme to come and bind, and it stops it prematurely to produce any kind of mRNA. So this is all about the attenuation. So in this video of tryptophan operon, we have talked about the tryptophan operon, what is an operon, how many structural genes are there, what are the functions of all these structural genes, how the structural genes are required in the formation of the tryptophan amino acid, how the tryptophan or trip operon is turned on and off, and how it is regulated by the attenuation mechanism. So this is all about the tryptophan operon. Thank you so much.